pathway. Ribose production, and ribose is very important. Used in transcription ribose and can be dephosphorylated deoxyribose DNA, and DNA very important DNA replication in cell division. Yes? So this pathway is very active in case of cancer. Very highly activated. Why? Because cancer uncontrolled, yes? Uncontrolled cell division. There is a requirement for cell division. For cell division, the DNA duplication, yes, must be versed. For duplication of DNA, the ribose required. For this pathway is very, very, very active in case of cancer. Yes? Okay, so very important, ribose. Number 2, NADPH. Provision of NADPH. And NADPH is very, very important reducing agent. Without NADPH production, the oxidized glutathione, which is inactive, cannot be converted to active glutathione. What is the function of glutathione? Antioxidant, anti-free radical. So glutathione protecting the cell membranes of red blood cells and hepatocytes and nephrons from oxidants or oxygen active species, and protecting the cell membranes from the free radical. So without the NADPH production, the patient will suffer from hemolytic anemia. The cell membrane of red blood cells will be distracted by oxidants, by free radicals. So glutathione, anti-cancer, anti-free radical, antioxidant. Yes. Okay. The main source of NADPH in our biological system is hexose monophosphate pathway. The major source, the main source of NADPH. Yes. Okay. Steps. So these are the main aims. But there is minor aims. Availability of glyceraldehyde triphosphate. But it is not the main aim. Yes? Because there are another sources for glyceraldehyde triphosphate. Yes? It's a glycolysis source of glyceraldehyde triphosphate. Okay, so hexose monophosphate pathway in islands, in liver, in kidney, in adrenal gland, yes, because the function of the NADPH not only reducing agent in activation of oxidized glutathione, but also as coenzyme for production of the hydrocortisone from cholesterol. Yes, where is the production? In which gland the hydrocortisone produced? Adrenal gland. Number 2, synthesis of testosterone. Testis. Ovary. Yes? Required for, so NADPH required for synthesis of the progesterone from cholesterol. So gonads. So this process is active in gonadals, adrenal gland, liver, kidney, but less active in the skeletal muscle. Whereas in the cells, in mitochondria or cell membrane and cell membrane. So hexose monophosphate pathway is occurring in the cell membranes, as in case of glycolysis. So number one, glucose will be phosphorylated to glucose 6-phosphate, glucokinase or hexokinase. So if the process occurring in the liver, glucokinase, yes? Other tissues, hexokinase. ATP to ADP, yes? Okay. Glucose 6-phosphate will be oxidized, yes, by glucose 6-phosphate dehydrogenase. NADP to NADPH. Yes? NADP and NADPH. So, the best, yes, required substance produced from the hexose monophosphate pathway is the reduced phosphorylated form of vitamin D3. Niacin, yes. Active form NADP, reduced form of NADP, NADPH. So it's a reducing agent, reducing equivalent. Reducing equivalent. 2, 6-phosphogluconolactone. 6-phosphogluconolactone. 6-phosphogluconolactone hydrolyzed to 6-phosphogluconate. 6-phosphogluconolactonase. 6-phosphogluconolactonase. 2,6-phosphogluconate. Yes? 6-phosphogluconate. 6 carbons, yes? 6 carbons, 6 carbons, 6 carbons, and 6 carbons. 6-phosphogluconate dehydrogenase. 6-phosphogluconate dehydrogenase. Dehydrogenase. NADP to NADPH. NADP to NADPH. Oxidative decarboxylation. Carbon dioxide. Yes? So, from hexose to pentose to 5 carbons, yes? Ribulose. Ribulose 5-phosphate. So therefore we called it pentose phosphate pathway. 5 carbons, pentose phosphate pathway. Yes, all the intermediates monophosphates. Therefore, we called it also hexose monophosphate pathway. Yes, to differentiate the glycolysis from hexose monophosphate pathway, in glycolysis, diphosphates can be formed. In hexose monophosphate, diphosphates cannot be formed. Only monophosphates. Yes, monophosphates compounds. Okay. So, in general, hexose monophosphate pathway can be classified into 2 phases. oxidative decarboxylative reactions or oxidative decarboxylation reactions, oxidative reactions, and it is irreversible. 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 Irreversible, irreversible, irreversible. Okay. Second phase, so this is first phase. A second phase is the non-oxidative reactions. Non-oxidative reactions. Yes, and it is reversible. Reversible reactions. The aim is of the hexose monophosphate pathway, its production of NADPH. Number 2, ribose. So the aim is, so we can get the aim is from the first phase. Yes. Second phase, it is just a reversible reaction. Ribulose will be isomerized by, isomerase. Isomerase. So keto ribulose can be converted to aldo ribose 5-phosphate, and it is a reversible reaction. Or by epimerase. Epimer of ribulose, xylulose. Xylulose 5-phosphate. Okay. That's all. 5 plus 5. 5 carbons plus 5 carbons. Yes, xylulose interacted with the ribose. 5 carbons with 5 carbons. 7 carbons plus 3 carbons. 3 carbons, aldo. 7 carbons, keto. Pseudoheptulose. Hepta. So, pseudoheptulose. Yes? Is lesser aldehyde. Aldo interacted with keto. Yes? Transketolase. Transketolase. Yes? Transketolase. Vitamin B1 required. BBB. Sorry, TBB. Thiamine pyrophosphate. And this reaction is used for estimation of the activity or presence or absence of vitamin B1. So how we can analyzing the availability, absence or presence of TBB by estimation the activity or the transketolase. So this reaction used in medicine for diagnosis of the vitamin B1 deficiency. Okay, في الكت ايش بيعمل؟ Yes. Okay. C7 plus C3 pseudoheptulose interacting with the lesser aldehyde. Reversible reaction. 2 C4 plus C6 fructose 6-phosphate erythriolose 4-phosphate keto, erythrose aldo keto. Okay, then C4 erythrose plus hexylulose C5 reversible reaction. C3 aldose glyceraldehyde triphosphate and C6 fructose reversible reactions وهكذا. Okay, so هذه هي الهكسود monophosphate pathway divided into two phases: irreversible oxidative reactions and reversible non-oxidative reactions. The aim of this pathway is production of the pentoses. Yes, pentoses. Ribose, deoxyribose. Yes, ribose used for synthesis of nucleotides. Yes, guanosine triphosphate. Yes, cytidine triphosphate. Yes, uracil triphosphate. Okay, so these nucleotides use it for what? So the ATP is adenosine triphosphate. So chemical structure for adenosine triphosphate, it is the nitrogen-based adenine and sugar, ribose, and the triphosphate, adenosine triphosphate. Cyclic adenosine monophosphate, the chemical structure of cyclic adenosine monophosphate is adenine and ribose and phosphate group. So regulatory or secondary messenger, cyclic adenosine monophosphate. Cyclic adenosine monophosphate. The chemical structure of NAD, nicotine amide dinucleotide. There is a ribose in the structure of NAD. Ribitol. What is the ribitol? Ribitol is the reduced form of ribose. Ribitol in chemical structure of fat, vitamin B2. Yes, so the ribose is important. Yes, so it is the chemical constituent of coenzymes, NAD, yes, ATP, synthesis of nucleotides, yes, so very important, ribose. And without ribose, the replication cannot be started. Why? Because the nucleotides are substrates for the replication. Yes, cell division. Okay. Second aim, it is the production of reduced form of NADP, NADPH, and NADPH, very important coenzyme, and synthesis of fatty acid, and synthesis of cholesterol, and synthesis of 6 hormones, male or female 6 hormones, testosterone, progesterone, estrogen. Yes. Okay. Synthesis of phospholipids. And NADPH also very important in detoxification. How? Because NADPH is the reducing equivalent required for reduction of oxidized glutathione to reduce the glutathione. Reduced glutathione is the active form of the glutathione. So can reduce the oxidants, oxidative reagent, like hydrogen peroxide, H2O2. Hydrogen peroxide produced normally from metabolism in our biological system, but it is toxic compound, must be reduced to water and oxygen. Therefore, any inhibition for oxidative phase of hexose monophosphate pathway will lead to hemolytic anemia. Yes, hemolytic anemia. How? Because NADPH produces. NADPH produces neutralizing hydrogen peroxide. How? We have two forms of the glutathione. GSSG, oxidized glutathione. Inactive by glutathione reductase converted to reduced glutathione. But the reduced equivalent required NADPH to NAD 2 GSH2. Yes? So it is active glutathione. The active glutathione, yes, it is responsible for hydrolysis. If it is H2O2, it is oxygen active species or hydrogen peroxide to water and oxygen inactive. Yes? Non-toxic, toxic. This compound produced from metabolism in our biological system, so must be neutralized. If the glucose 6-phosphate inhibited or mutated, so NADPH absent, the glutathione oxidized inactive cannot be converted to active. The produced in our biological system